Hey everyone, welcome back to another topic review video for Bio in a Bottle. I'm Ruben, and today we're going to go over both topic 1.4 and 1.5. I had previously mentioned certain biological macromolecules, nucleic acids, proteins, carbohydrates, and lipids. But in this video, we're going to be going over both the structure and the function of these macromolecules. We can begin with amino acids by going through the process of protein folding. Proteins have a ton of roles within our body, and they're used all the time. However, a protein doesn't just come out of nowhere, and there's a specific process which creates these complex proteins that have specific functions. This process is called protein folding. There are four structures within protein folding: primary, secondary, tertiary, and quaternary. Primary structure is the first structure a protein begins, which is a chain of monomers of proteins, which are called amino acids. Within each of these monomers, there's an amino group, carboxyl group, and an R group. Each of these components play a role within the amino acid. The amino group and the carboxyl group of the amino acid maintains the directionality of the amino acid, in which the carboxyl group of one amino acid can form a peptide bond with the amino group of a different amino acid, which allows these individual monomers to form a polypeptide chain when multiple are connected. Once a polypeptide chain is formed through the process of protein synthesis, the primary structure is complete, and the secondary structure is next. Within the secondary structure, the chain of amino acids folds into different ways, two of the most common being an alpha helix or a beta pleated sheet. The way it folds is based on how the different amino acids are arranged within the chain. The different types of amino acids are defined by the R group of the amino acid, as the R group can differ and have effects on the protein structure. An R group can be hydrophilic, hydrophobic, and ionic. The R groups have a further effect on the protein structure within tertiary structure, as hydrophilic amino acids will tend to be on the outside of the protein, while hydrophobic amino acids will tend to be on the inside of the protein. Quaternary structure is the final protein structure, in which multiple tertiary structure proteins connect through hydrogen or disulfide bonds. Not all proteins end up in this structure, though. Okay, well that's pretty much it for protein folding, which is the bulk of this video. Next, we can talk about nucleic acids. Nucleic acids, whether it's DNA or RNA, are made up of monomers called nucleotides. Within these nucleotides, there are three components: a phosphate group, a 5-carbon sugar group, and a nitrogen base. However, these components can differ based on whether the nucleic acid is DNA or RNA. First of all, the 5-carbon sugar group is different from DNA and RNA. The 5-carbon sugar group in DNA is deoxyribose, while for RNA, it is ribose sugar. Also, while both DNA and RNA share the nitrogen bases adenine, guanine, and cytosine, DNA can have a thymine base, something not found in RNA, and RNA can have a uracil base, something not found in DNA. DNA and RNA also have more differences in its structure, as DNA is double-stranded, while RNA is single-stranded. DNA is anti-parallel. This means that individual strands run opposite of each other. The directionality of DNA strands are defined by its 5' and 3' ends, as labeled here. How do these strands connect, though? They connect through the nucleotide regions of each strand, in which specific nucleotides will pair together. An adenine nucleotide will pair with a thymine nucleotide with 2 hydrogen bonds, and a cytosine nucleotide will pair with a guanine nucleotide with 3 hydrogen bonds. Next, we will be talking about carbohydrates. The type of carbohydrate depends on the structure of the monomers, in which its arrangement will determine the function of the carbohydrate. Carbohydrate monomers, known as monosaccharides, connect through covalent bonds. When two monosaccharides connect, they will form a disaccharide, and when many connect, they will form a polysaccharide. Polysaccharides can either be a straight chain of monosaccharides, but they can also be branched. Finally, we will be going over lipids. Lipids are nonpolar, which means they are hydrophobic. There are different structures and functions for lipid. This is a phospholipid, in which contains a polar head, which is hydrophilic and faces an aqueous environment, and a nonpolar tail, which is hydrophobic and does not face the aqueous environment. That's about it for this video. Thanks for watching, and make sure to subscribe for more AP Biology content.